Hello, welcome back to my channel. Today we'll discuss about immunoglobulins. Immunoglobulins, they are a heterogeneous group. They are a heterogeneous group of specialized serum proteins. Specialized serum proteins that are formed in response to an antigen and they react against the same antigen or a similar one in an observable manner. They react with the same antigen or a similar antigen in an or a molecule in an observable manner. Then they constitute around 20 to 25% of the serum proteins, and they are produced by plasma cells. Coming on to the first immunoglobulin, that is IgG. Okay, this is the most abundant immunoglobulin and constitutes around 80% of the total immunoglobulins. So this is present in large quantities or amounts, you can say. It is also referred to as a versatile molecule or a general purpose antibody. Because it protects against the diseases or the infections of both blood as well as tissue. Okay, and then in IgG we have the coefficient of sedimentation as 70s. It appears late, but it will persist for a longer period of time. It appears mainly after IgM reaction. Okay, then coming on to it will activate both the pathways of the complement system. I'll be discussing complement system also, not in this video but in the other. Then it is the only immunoglobulin. This is a very important point. It is the only immunoglobulin which can cross the placenta and then confer the immunity from the mother to the fetus. So only immunoglobulin that can cross the placenta, correct? Now coming on to the functions. When you mention immunoglobulin, you have to mention first the properties and then the uses, the functions. So in functions, first of all, it confers the immunity to the fetus, from mother. Then increases phagocytosis of microorganisms, induces or increases the phagocytosis of the microorganisms. It also helps the NK cells. NK cells in finding target cells. Then there is a coating formed over it, due to which they are then attacked by the NK cells and further the reaction is done. Then in the fourth point, IgG, it is also responsible, since it activates both the complement pathway, so it has a bigger role in conferring the immunity. Then coming on to, it is also responsible for participating in various immunological reactions, that is the antigen antibody reactions like neutralization reaction, immunological reactions like CFT or neutralization test, etc. Then coming on to the fifth, it is also responsible for the it can also immobilize the bacteria. It can immobilize the bacteria by binding to the cilia or the fimbriae, by binding to cilia or flagella. Then it can also be passively administered IgG. Then it will suppress the homologous antibody synthesis by a feedback process. We have seen that in passive immunization we usually give antibodies. So when IgG is administered passively, it impedes or it suppresses homologous antibody synthesis by feedback process. The structure of IgG antibody or immunoglobulins, the structure of IgG. So it is a Y-shaped molecule. These will be linked by sulfide linkages. Here we'll have double sulfide or disulfide linkages. Okay, this is the Fab fragment. This is the FC fragment. Then this is the heavy chain. This is the light chain.